Hello friends and welcome to my channel Sequential Biology. In this video we will discuss fertilization process in sea urchin and fusion of male and female gamete followed by the union of their pronuclei to form the diploid zygote is called as fertilization. But before going to discuss about detailed process of sea urchin fertilization, first we should have clear concept about the structure of sea urchin egg. Sea urchin is a species of phylum Echinodermata, and in sea urchin egg you can find different type of membrane like any other egg of different species. Okay, so outermost layer is called as egg jelly, and inside this egg jelly and another important layer is present which is called as vitelline membrane, and inside this vitelline membrane the egg membrane are present, and in egg membrane different type of receptor are present and which are known as receptor of egg membrane. And this egg membrane cover the protoplasm, and protoplasm is made up of cytoplasm and nucleus, but this nucleus is not fully formed and this is called as female pronucleus. And in cytoplasm you can find different type of structure. The mitochondria can be found in cytoplasm and also the protein droplets can be found in cytoplasm. And since their egg is microlithial, so few amount of the egg granules can also be found in their cytoplasm. Ultimate target of fertilization is union of male and female pronuclei. First we will see how these male and female pronuclei will be united. Sometimes you become confused about this process, and here is a clear-cut diagram about union of male and female pronuclei. An egg you can find egg pronucleus, but the sperm, when the sperm membrane fused with egg membrane, the sperm released not only their pronucleus but also they release centrosome, right? So this is the centrosome of sperm and this is sperm pronuclei. And just after releasing of the centrosome and male pronuclei, the nucleus and centrosome will rotate 180 degree in egg cytoplasm. Just after releasing of this male pronuclei and the centrosome, you can find the male pronucleus just in front of the centrosome, but these two structure rotate 180 degree, so you can find this centrosome in front of pronucleus, male pronucleus. Then the nucleus plus cytosome rotate 180 degree in egg cytoplasm, so centriole becomes in front of the nucleus between egg and the sperm pronuclei. Now the polymerization of the microtubule in the centriole will form the sperm ester, and through this sperm ester the male pronucleus move towards the egg pronucleus, and ultimately male pronucleus and egg pronucleus will be united to form a diploid zygote. Then the sperm entry stimulate the egg or the secondary oocyte and the meiosis II will be completed, and the haploid mature ovum and the second polar body is formed during meiosis II cell division. The chromosome of the ovum are not organized into a nucleus, and this unorganized nucleus is called as female pronucleus. Only the centrosome and the male pronucleus enters into the egg. So only the centrosome and the male pronucleus enters into the egg. Sperm ester reaches the female pronucleus, then the female pronucleus move along the rays towards the male pronucleus, right? So both pronuclei pushed to the center of the egg by expansion of the sperm ester, and their membrane fuse and resulting the formation of a single membrane which encloses both male and female pronuclei called the pronuclear fusion, and ultimately this will causes the formation of a diploid zygote. Then this is egg centriole and this is sperm centriole, and ultimately the sperm pronuclei and the egg pronuclei will be united to form the diploid zygote, and the union of these two pronuclei is called as pronuclear fusion. Now we will see the different step of fertilization in sea urchin. The sea urchin is a model animal of developmental biology, and usually the fertilization process is studied in this species because the gametes are very easy to collect and the fertilization is external, and fertilization provides a good model because the same process occurs in case of vertebrates, and the researchers can observe the fertilization and subsequent developmental process simply by combining eggs and the sperm in seawater in lab. And for this reason usually the fertilization process is studied in sea urchin. So sea urchin is a model organism in developmental biology. The first step of their fertilization is contact and recognition between sperm and egg. Because the fertilization is external, so it is very much important for species specific recognition of sperm and egg. A resect, a 14 amino acid peptide found in egg jelly of sea urchin, and just outer layer of their egg is called as egg jelly, and this egg jelly contains a resect. Resect, a 14 amino acid peptide, this is a very small protein found in egg jelly of the sea urchin Arabasia punctulata, and the resect diffuse in sea water. So this is the resect, and these are found in egg jelly, and after releasing of this egg in the sea water, this resect will be diffused in the sea water, right? So resect diffuse in sea water, and very low concentration attract the sperm of its species. So the sperm are also released in the sea water, and this sperm will be attracted towards this resect, and these resect are found just nearer to the egg. Then the resect binds with the sperm receptor, and this resect binds with sperm receptor. In the green color I have drawn the sperm. So the sperm receptor are present in their membrane, the sperm membrane, and the resect binds with this sperm receptor, and so calcium enters into the sperm and sperm moves towards the egg, and this is called as contact and recognition between sperm and egg. Alright, this is the first step of their fertilization. Various scientists proposed various factors that attract the sperm towards the egg. According to F.R. Lilly, the fertilization, which is a type of glycoprotein or the mucopolysaccharide from the egg jelly and the egg membrane, and the anti-fertilization from the sperm. Then the fertilization can be found in egg jelly, and the anti-fertilization can be found in sperm. And the Rothschild told that the gynogamon 1 and 2 can be found in egg jelly, and the androgamon 1, 2 and 3 can be found in sperm. These are the different idea about how the species specific recognition of the sperm and egg can be occurred. Next step is called as acrosomal reaction. Exocytosis of the sperm acrosomal vesicle released hydrolytic enzyme make a hole in egg jelly coat, and just outer layer of their egg is called as egg jelly, and acrosomal enzyme causes breakdown of this egg jelly coat, and acrosomal process covered with the protein then bind to the receptor in the egg plasma membrane. Just after the egg jelly there are present egg plasma membrane of the vitelline membrane, and the vitelline membrane or the plasma membrane of the egg have the species specific then receptor. Fucose sulfate from the egg jelly binds with sperm receptor, and influx of the calcium. So inside this sperm the calcium level will be increased, and another important type of second messenger will be increased inside the sperm, and which is known as IP3 or inositol 1,4,5 triphosphate in the sperm. And the calcium binds with acrosomal vesicle, and the fusion between the acrosome membrane and the sperm membrane, and the G-actin polymerase to form the F-actin, and this form the acrosomal process. And the acrosomal hydrolytic or the proteolytic enzyme digests the jelly coat, and also the fertilization cone protrude from the sperm head penetrate through the egg jelly coat, and the fertilization cone penetrate through the vitelline membrane due to the digestion of the acrosomal enzyme, and ultimately binds with bindin receptor in egg membrane. Then what will be the diagram of this total process? You can see the plasma membrane. Just outside of the plasma membrane there are present the vitelline membrane, and outside of this vitelline membrane the egg jelly are present. Jelly contains the fucose sulfate, and in the sperm membrane there are present fucose sulfase receptor, and this is the sperm head, and in this sperm head you can find the acrosomal vesicle, and just below this sperm head you can find the nucleus, and in between the nucleus and the acrosome there are present G-actin monomer, and this G-actin polymerase to form the cone, right? Now just after binding of this fucose sulfate into the sperm receptor, the calcium ion enters into the sperm head, and so the acrosomal membrane will be fused with the sperm membrane, and this causes the release of this acrosomal content, and this acrosomal content contains the acrosomin. This is a type of protease and the lysine, and this causes the breakdown of this egg jelly layer, and ultimately just inside of this acrosomal vesicle you can find the G-actin and the G-actin polymerase, and this will form the actin filament, and this actin filament form the fertilization process, And in front of this fertilization process, the bindin can be bound, right? So these are the bindin, and these bindin are present in this fertilization process. And in plasma membrane, there are present bindin receptor. So these bindin receptor are present in the plasma membrane of egg, and the bindin are present fertilization cone of sperm. Remember it, the bindin is present in fertilization cone of sperm, and the bindin receptor are present in the plasma membrane of egg, right? So after binding of this bindin with this bindin receptor, then the sperm break this vitelline membrane, and ultimately the sperm membrane fused with the plasma membrane of egg. And the vitelline membrane, tough non-cellular layer present between the egg jelly and the plasma membrane, and made up of glycoprotein and the polysaccharide. Now the binding of the sperm to the egg membrane or formation of the fertilization cone, the lysis of the vitelline membrane allow the fusion of the sperm plasma membrane with the plasma membrane of egg. So several microvilli in the egg plasma membrane which engulf the head of sperm, right? So these are the several microvilli, and this engulf the head of sperm, and this head of sperm contains the nucleus and the centrosome. Then the several microvilli in the egg plasma membrane which engulf the head of the sperm, called the fertilization cone, or extended by the polymerization of the actin. And the secreted content of the cortical granules also clip up the of sperm binding receptor. And next process is prevention of polyspermy. Then the cortical granules will be released in the perivitelline space. Now the sea urchin is monospermy. That means only one sperm enters the egg, and the entry of many sperm may establish the polyploidy. In sea urchin, the two blocks to avoid the polyspermy can be found. One is the fast block of polyspermy, and another is slow block of polyspermy. In fast block of polyspermy, the temporary electric change in the egg plasma membrane can be seen. And usually inside the egg plasma membrane, the charge is negative, right? So the temporary electrical change in the egg plasma membrane from negative 70 millivolt to the positive millivolt, and since the sperm membrane is also positive, so the repulsion can be occurred, and this will causes the prevention of polyspermy, and this is called as fast block to polyspermy. Sperm fast block, the slow block of the sperm can be seen, and this is called as the permanent block, and occur due to the cortical reaction. Inside of the plasma membrane, the many cortical granules are present, and these cortical granules will be released in the perivitelline space, and this will causes the formation of fertilization membrane, and this also prevent the polyspermy. Then the two types of prevention of polyspermy can be seen in case of sea urchin. One is fast block and another is slow block to polyspermy, right? Fast block polyspermy remains only few minutes. In sea urchin, egg remains positive only for few minutes, so it is not sufficient to prevent the polyspermy, hence the slow block is needed. So fast block of polyspermy, how this is occurred: start within 1 to 3 seconds after binding of the sperm, and then last for 60 seconds. So this is the first sperm, and first the egg plasma membrane resting potential is minus 70 millivolt, and the sperm binds with the egg at the resting membrane potential minus 70 millivolt, but cannot bind with the membrane of the positive charge. Just after binding of the first sperm, the small influx of the Na plus into the egg. So the small influx of Na plus into the egg, since this is the positive charge, so the membrane potential shift to the positive level, that is the plus 20 millivolt. Except the first sperm, the other sperm will not be attached with the plasma membrane, and this is called a slow block of polyspermy. Because of the sperm carry a voltage sensitive component, that is the positively charged fusogenic protein. Sperm carry a voltage sensitive component which is called as positively fusogenic protein, and also the egg membrane also becomes positively charged, so a repulsion can be seen, and this is called as fast block to polyspermy. And the slow block to polyspermy occurred by cortical reaction. The fusion of the sperm and the egg membrane activate IP3 and the DAG. These are a kind of second messenger, and the calcium released from the calcium storage of the egg, and the calcium fuses with the cortical granules with the egg membrane. Okay, so these cortical granules are present beneath the egg membrane before fertilization, and the cortical granules contain the following substances released between the vitelline membrane and the plasma membrane. And these cortical granules contain the trypsin-like protease that dissolve the protein coat that connect the vitelline membrane to the plasma membrane. And also these cortical granules contain many mucopolysaccharides. This create osmotic gradient water pushed between the vitelline membrane and the plasma membrane. So vitelline envelope expand and form the fertilization envelope. And also the peroxidase enzyme can be found, and this hardens the fertilization envelope. And the hyaline component also found. This form the coating around the egg below the fertilization membrane. This is the vitelline membrane and this is the plasma membrane, and this portion you can see, this portion is before fertilization. And before fertilization the cortical granules are present just inside of the plasma membrane. Binding of the first sperm with the egg membrane, the IP3 and the DAG. Second messenger IP3 and the DAG will be synthesized, and this will act on the smooth endoplasmic reticulum of egg. And you know the smooth endoplasmic reticulum is calcium storage organelle, and this calcium binds with these cortical granules, and so these cortical granules will be fused with egg membrane, and the content of the cortical granules will be released in between vitelline membrane and the egg membrane or the perivitelline space. And this cortical granules contains different type of component. The trypsin-like protease clips up the bindin receptor. So this is the bindin receptor, and the trypsin-like protease clips up this bindin receptor. And fertilization is not only causes the formation of the diploid zygote, but also this fertilization activate many metabolic process of egg. Sea urchin egg sluggish and the egg is activated by the sperm. Activation start with the two block of the polyspermy. And the first block is influx of Na+, so Na+ ion will be increased inside the cell, and the slow block causes the increase of intracellular calcium level and 3 to 5 fold increase in the oxygen consumption, and so that the NAD+ will be converted into the NADP+, and this biosynthesis of the new membrane of the lipid, and during the time of cleavage the membranous component must be synthesized, and this is occurred due to this first block and the slow block of polyspermy, because the NAD+ will be converted into the NADP+ by a special kind of enzyme which is called as NAD+ kinase enzyme, and this causes the biosynthesis of new membrane lipid, and this will help in the next stage, that is the cleavage process. And the union, and after then the union of male and female pronuclei can be seen, and we have already discussed about this union of this male and the female pronuclei. Then these are the detailed process of fertilization in sea urchin, and hope you have understand the detailed process of this fertilization in sea urchin, and in the next video we will discuss the cleavage. So thank you, and that's it.